For antibodies to be produced in the body to fight against infections, there must be a very important event that occurs, and that event is called B cell development. So the B cell development are in stages, all right, beginning from the stem cell, which is one of the major cell that differentiates into different cells, whether it's coming from the myeloid pathway or the lymphoid pathway. So from the stem cell, you have differentiation, and the stem cell give rise to all these stages until we get to the mature B cell. So we are going to be walking through these stages and what happens in these stages. All right. Now, if you observe, you see this big cell here. Now, this big cell is called the bone marrow stromal cell. Stromal cell. So this stromal cell is found in the bone marrow, specifically the endosteal niche. The endosteal niche of the bone marrow. Now, this cell, the binding between this cell and the stem cell factor begins the process of development and maturation of B cells. All right, let's start from the first one, which is the stem cell. Now, there is a binding between two proteins, one from the bone marrow stromal cell, that is called the VCAM1. So there is a binding between the VCAM1 and another protein on the surface of the stem cell, and that protein is called the VLA4. Okay, and there is a binding between VCAM1 and the VLA4. Now, when this binding occurs, there is a heavy chain rearrangement. Okay, now that means that the DNA transcribes and translates the heavy chains. So there is a heavy chain rearrangement, and that heavy chain rearrangement, the first one is the D heavy chain and the J heavy chain. So there is a D and J heavy chain arrangement. Now it progresses to the next phase. Now the next phase is the pro B cell, which is made up of the early B cell, early pro B cell rather, and the late pro B cell. Now what happens in the early pro B cell is that there is also a binding between a protein coming from the stromal cell and that of the early pro B cell. And that binding is between what we call the stem cell factor and c-kit. All right. Now there's a binding between the stem cell factor and c-kit. Now what does this binding do? It progresses this early pro B stage to the late pro B stage. Now, when that happens, there is a complete heavy chain rearrangement, which is the V heavy chain, D heavy chain, and J heavy chain rearrangement. Okay, now the heavy chains are complete. Now it progresses to the next stage, which is the late pro B cell. Now this binding enables the early pro B cells to present what we call an interleukin receptor on their surface. Now, an interleukin receptor is there to receive an interleukin. An interleukin is a chemokine that is produced by the stromal cell. So we have chemokines produced by the stromal cell. So the stromal cell synthesizes the interleukin 7. So it synthesizes interleukin 7 that binds to the interleukin 7 receptor on the late pro B cell. So that is that for that event. Now, the moment that happens, there is a downregulation of the stem cell factor and the c-kit. Now, this enables it to detach from the stromal cell. Okay, still in the bone marrow, but it detaches from the stromal cell. Now, this takes us to the next stage, which is the pre-B cell stage. Now, in this stage, something happens. These heavy chains that has been transcribed and translated are now presented, are brought to the surface of the pre-B cell, and they now form what we call the heavy and the surrogate light chain. So we have the heavy chain, these are the heavy chain, and these are the surrogate light chain. Okay, now these are formed, and this is also called the pre-B cell receptor. Okay, now this acts as a receptor, but it's a pre-B cell receptor because it is formed on the pre-B cell. Now, at this point, another recombination of genes occur. Now, that is what we call the V and J heavy chain, light chain rather, V light chain and J light chain. Now, this forms the light chain, replacing the surrogate light chain. Now, moving to the next phase, which is the immature B cell, something happens. This pre-B cell becomes an original B cell, okay, having both the heavy chains and the light chains. Now, this is now called the B cell receptor. It is no longer the pre-B cell receptor, it's now called the B cell receptor. Okay, so that is the immature B cell. Now, that ends the stage in the bone marrow. So we can now see that the B cell actually does the chunk of development in the bone marrow. However, it moves out from the bone marrow to secondary lymphoid organs, which are maybe the spleen and the lymph nodes, to complete its development. So what happens is that the B cell moves from the bone marrow to the secondary lymphoid organ. Now, at this stage, it is now called the naive B cell. And why is it called the naive B cell? It presents both types. Now, this is now an immunoglobulin. That is, this is now an antibody, right? Now, it presents both the antibody M, which is always the first antibody to be synthesized, and another type of antibody. We call that antibody D. Okay, now these are antibodies waiting for an antigen to bind. Remember, these other stages are in the secondary lymphoid organs. Now, the moment you have a binding of an antigen, so let's label this an antigen. The moment you have a binding of an antigen to the antibody or immunoglobulin, that B cell automatically becomes a mature B cell. And at this stage, the antigen is taken into the B cell, shredded into portions, and presented on a particular molecule, which I'm going to talk about in another video, for the T cell to recognize it and activate it for it to differentiate into the plasma cell and the memory cell. So the plasma cell in turn produces that same antibody that is produced as a result of the type of antigen that's attached to it. So that is very important. So this is the developmental stages of B cell.